Assalamualaikum and a very good morning to our lecturer Dr. Wan Nur Farahin binti Wan Osman and to all our fellow classmates. We are from Group 3 are going to present about the transplantation and graft rejection. Before we start, let me introduce our group members. My name is Munirah binti Sulhairi, followed by Sharifah Nur Izzati binti Said Mustafa, Muhammad Huzaifah bin Hassan, Muhammad Tarmizi bin Muhammad Nur, and Nur Atika binti Saiku Jalaluddin. First of all, I would like to start our presentation with the definition of transplantation. What is transplantation? Transplantation is a graft or organ transplant. It is a process by surgically we transfer or we remove organs, tissue or cells from one site or area of a person's body to another area of a person's body, or from a person known as a donor to another person known as a recipient. The purpose of transplantation is to replace or repairing damaged or diseased organ, tissue or cells. But some of the recipient will experience an organ transplanted failure to function. This is due to the transplantation rejection. Transplantation rejection is a process of transplant immune system recipient that attack the transplanted organ, tissue, or cells, and it will cause the destruction and system failure in functioning of the transplanted organ, tissue, or cells. Why is this happen? Our body immune system will usually protect us from any harmful substances such as poison, germs, and cancer cells. So these harmful substances contain protein known as an antigen that coat their surface. As soon as these antigen enter our body, immune system will recognize them as a foreign substances and will attack them. Same goes to when the person receives an organ from someone else during the transplant surgery. The person's immune system will recognize that as a foreign substances. This might be due to the immune system detect antigen on the cell of the organ are not matched. Let's move to the importance of transplantation. Transplantation is very important as the treatment for the end stage of the organ failure, such as heart and liver failure. It can provide the chances for the patient to survive. Solid organ transplant will increase the life expectancy and improve the life quality of a patient. For example, the tissue and corneal transplant from the donor can help the recipient to see clearly, move better, and lead to a better life. Now, what are the symptoms of transplantation rejection? First, the organ's function may start to decrease and fail to function. Second, the recipient will experience the discomfort and uneasiness at the area of transplanted organ. Second, might also occur pain or swelling around the area of transplanted organ. And lastly, they will also experience fever, flu-like symptoms including chills, body aches, nausea, cough, and shortness of breath. That's all from me. I will pass my presentation to Sharifah Nur Izzati. Thank you.
Okay, thank you, Munira. So now I'm going to continue with types of transplant. The first one is the autograft. Okay, autograft ni dia adalah transplant of tissue from one side to another within the same person. Maksudnya dekat sini contoh, seorang tu dia accident and dia injured dekat dia punya peha. Then to replace the tissue, kita boleh guna tissue dekat another part of his ataupun her body. The example, contoh dia macam vein and juga stem cells. Second, the allograft. Allograft adalah human to human. Basically, the definition is that the transplant of an organ or tissue between two genetically non-identical members of the same species. Maksudnya, dia berlaku transplant antara spesies yang sama tapi orang yang berbeza. Okay, sebab dia genetik yang berbeza. So, this is common lah, like kidney transplant. And this is the example of autograft and juga allograft, which is the osteochondral graft. It is a replacement core made of cartilage and bone from another site in the knee, or a tissue donor knee is then made to fit into the hole. Kat sini maksudnya kalau autograft tu dia boleh replace cartilage dengan bone tu dia guna another side dekat dia punya knee, ataupun kalau allograft dia akan ambil tisu donor punya knee lah. Alright, so now is the xenograft. Xenograft adalah human to animal transplant. Okay, sebabnya dia berlaku across species barrier. One of the successful xenograft transplant is the heart valve transplant of porcine. Porcine is type of pig. Okay. However, since that human dengan animal dia punya macam immune response, dia punya organ tak sama, so it has a high risk to cause some complications such as the rejection disease or non-compatibility in blood. Okay, last one is the isograft, also known as syngeneic graft. It is the human to human juga, but the difference with the allograft is that dia melibatkan identical twin. Okay, the transplant of an organ or tissue between two genetically identical twin of the same species and does not trigger immune response. Why dia tak trigger immune response? Disebabkan dia ada genetik yang sama. Okay, contoh dia macam bone marrow, disebabkan dia ada histocompatibility antigen adalah sama, jadi the graft survive and not rejected. Okay, this is the last slide. Okay, it is just to sum up all the things about the types of transplant. The first one is autograft, which occurred within the same individual, syngeneic ataupun isograft, antara identical twins, allograft, non-identical individual, dan juga xenograft between different species. So that's all for me. I pass my presentation to Tarmizi.
Okay, guys, I will continue with the component involved in the transplantation rejection. The first one is B and T cell. Second one is human leukocyte antigen (HLA). HLA system responsible for the encoding MHC in human. For example, MHC class 1 encoded by HLA A, B, and C. The third one is APC, antigen presenting cells. For example, macrophages, dendritic cells. And the last one is cytokines, for example, tumor necrosis factor. So as we all know, the heart of immune system is the ability in distinguishing between self and non-self. So all the nucleated cells in our body possess a unique measure histocompatibility complex (MHC) that function as identification tag. So different organisms have distinct self marker that prevent the transplantation unless from very close donor. So while the antigen that recognized as foreign to our body will stimulate the lymphocyte to produce antibody which specifically bind to the epitopes. That is why there are many cases of graft rejection caused by the self marker from the other organism. So, however, when we dive into the blood, luckily our blood system has their own antigenic system. So, ABO group incompatibility is a barrier solid organ transplantation because this antibody can bind to the respective antigen, and this binding will activate the complement cascade, will leading to the hyperacute rejection. I think that is all from me. Thank you.
Thank you, Tarmizi. Okay, I'm Huzefa, and I'm going to continue the other 2 points for immunology of rejection, which is transplantation antigen and mechanism of transplantation rejection. For the transplantation antigen, it consists of MHC, or also known as major histocompatibility complex. There are 2 classes, which is class 1 antigen and class 2 antigen. Class 1 antigen only express class 1 antigen will express on most of cells, but class 2 antigen only express on the cells of lymphoid system. MHC were gene complex which encode polymorphic cell surface glycoprotein involved in antigen recognition and antigen representations. So next is mechanism of rejection. Mechanism of transplantation rejection, which involve both cell-mediated and antibody-mediated hypersensitivity reaction directed MHC on foreign graft. There are 2 pathways, which is directed pathway and indirect pathway. For direct pathway, we initiate by APC that express the class 1 and class 2 MHC molecules. For the class 1 MHC molecules, will be detected by CD4, CD8+, also known as cytotoxic lymphocyte, which is it activate and duplicate and attack the foreign organs. The attacking process will cause the cell to release cytokines, and it will lead to inflammation to be happen, and also can cause the plaque formations that block the circulations of blood and cause the ischemia. And for indirect pathway also involved with APC, which is the MH2 class molecule that express or detected and activate the CD4, which also known as T helper cells. These T helper cells can be go into two pathway, which is activate the B lymphocyte when the graft antigen were detected, and the B lymphocyte will turn into plasma cells and cause it to produce antibody. And the other pathway, which is T helper cells, will produce interferon gamma interferons that activate the macrophage and may lead to ischemic. So that's all for me. Thank you. I will pass to Atika for explanation on stage of rejection.
Next, I will present about the clinical stages in transplant rejection. So there are three stages, which is hyperacute, acute, and also chronic. So for the first one, the hyperacute rejection, it occurs within minutes or hours after the transplantation due to the vascularization damage. The damage is caused by massive thrombosis by activation of the complement system. It is humoral mediated due to presence of pre-existing antibody of the recipient that match with the donor's antigen. It can probably happen due to the previous blood transfusion or multiple pregnancies. So the antibody will react with the cells inside the graft's blood vessel and causes a blood clot to form. This will eventually prevent the blood supply from reaching the graft and known as immediate rejection. So kidney is one of the most susceptible organ for hyperacute rejection. The next one, for acute rejection, it occurs within days or weeks after transplantation. Usually, it is for the first 6 months of transplantation. So, it is mediated by IgG antibodies. It has two types, which is acute cellular rejection or known as T-cell mediated, and another one is acute antibody mediated rejection. For acute cellular rejection, so the activated CD4 plus T-cell will secrete cytokine. And this cytokine will activate the macrophage and lymphocyte. As a result, there will be inflammation inside the graft due to the massive infiltration of these macrophage and lymphocytes. Besides that, there will be increase in vascular permeability and also local accumulation of mononuclear cells. As for the acute antibody-mediated rejections, anti-donor antibodies will be produced after the transplantation. So the antibody produced will cause microvascular endotheliasis and interstitial inflammations. And the last one, which is chronic rejection, it will occur within months or years after transplantations due to scarring of tissue or organs. So it is caused by both antibody and cell-mediated immunity, and it will result in inflammation, smooth muscle proliferation, and also fibrosis. So the major cause of this chronic rejection is due to previous episodes of acute rejections, inadequate supply of immunosuppressions, and also post-transplantation infections. That's all from us. Thank you.